slim, and these are automatically segmented. This is machine learning. We are applying some algorithms that come from the Kinect that we were talking earlier. So you know that the Kinect is doing machine learning, where it's recognizing your body postures during dancing. And the algorithm they are using is called random forest, which basically, I'm no expert on that at all, so I'm working with Min Do, who is an expert on this, a professor in my department, and computer science graduate students. But the idea is that the training takes a long time. So for the Kinect in particular, what they did probably, they trained that on millions and millions of kids dancing. But the testing is almost instantaneous. So the Kinect is recognizing your body position at 200 frames a second. But it's all based on those millions and millions of. So we're trying to kind of do the same thing. So take a long time to teach the computer how these things are looking like, slim images. And hopefully at the end have a very fast kind of testing. Here is where we are. So segmenting is the first problem. So simply taking the gland out of the stroma, that sounds simple enough for a human eye, not so simple for the computer. As you know, finding the dog in the picture is still a challenge. So it works very well for the low grades. That means when those glands are well defined. When the glands start to merge with one another, you can tell that that's going to be a problem. So that's why our area under the curve for 5 plus 5, these glands are disappearing essentially. So that's why our accuracy is not so good. But overall, I think it's not bad. So it's so around here in the middle, it's 96%. That's pretty good. So here's the first result on distinguishing between grades 3 and 4. So remember, this is that turning point where you go to aggressive treatment if you're 4, or you stay with active surveillance at 3. So depending on which groups of data we're looking at, we're somewhere at 75%, average 76%, distinguishing between the two. Now, again, I want this to be higher than this, and we cannot help but think sometimes that actually our gold standard is not perfect. The gold standard is the human. And it's not only one pathologist, but in this particular case, it's actually our gold standard is a consensus of 3. Now, there are studies that show that if you give the same samples to 3 pathologists, they agree exactly on 75% of the cases. So there might be some of that variability built in here. I consider it a good first step. So I think this is still publishable the way it is. But we're trying to see whether we can boost this up. So it would be very nice to have this automatically done. In terms of screening, so we're thinking that it could be very useful to have the bulk of the diagnosis, automate that. So let's say a computer that has 80% of the slides in a clinic, I think that's doable. And then raise flags for the pathologists for those more complicated problems. So in that case, a binary problem, one marker that's very interesting is these epithelial cells right here. So these are called basal cells at the base of the gland. If they are present in a nice single layer like this, that's a sign of a benign gland. When they are not present like this, that's a bad sign. That's basically tumors. So just looking for that, actually, we started to implement that into the machine learning. So now you see the different kind of segmentation. And basically, we're putting all this in is a histogram of all the features per pixel. And basically, at the end of the day, what we get where we are so far is this. It's kind of a nice separation, actually, between normal in green, cancer in red, and HGP. This is a high-grade prostatic intraepithelial neoplasia. So this is a new growth. It's not yet cancer, but it may become cancer. So it's an abnormal growth. Okay? It may look like cancer, but actually it's benign. But it has to be monitored. So it's kind of interesting, and it was the pathologists were excited that we're kind of able to distinguish between HGP and normal. So it's not great fun. So at least between normal and cancer, I think this could be, as I said, somewhere 80% accuracy to just screen through most of the cases and refer the complicated ones to the human, to the pathologist. Okay, so this is where we are. As I said, we're learning that it's a lot harder to publish in these clinical journals when you have to image hundreds and hundreds of samples. We're now doing validation 2 or 3 times on thousands more. So that's going to be a slow process. But hopefully in the end, the impact is also kind of correlating with that. So it may translate into saving a life or two, hopefully. We're also working on breast cancer, so we're trying to recover the results from prostate on breast. I'm not going to talk about this. This is just a kind of preliminary paper where we tested actually the pathologists. We trained them for 10 minutes, 3 different pathologists. We gave them the H&E and then here's the slim on 10 images. Look at both, you have 10 minutes. And then we fed them 100 just slim images and asked them to diagnose them. And they weren't as good as with H&E, but not much worse. So there were a few percentage points below the H&E with 10 minutes of training. On H&E, they got 6 years of training. So basically, that proves that the structure information is there for diagnosis, therefore we can move forward and kind of do what we did in process. Okay, let me move to a completely different application. And that is cell dynamics. So some of you may know that actually, if you measure the phase through an object, and let's say there is water coming in and out of the cell, let's say there is water coming out of the cell. So it's under osmotic stress. We're able to show back here at MIT, back in 08, that because there are these two phenomena that happen, the water coming out tends to reduce the thickness of the cell. On the other hand, the refractive index inside the cell goes up. It's like the cell becomes more concentrated because the water came in. So we were able to show in our calculation, we used the scattering model, that actually these two phenomena cancel out in the first order. Meaning that if you have small amounts of water coming in and out of the cell, if you integrate the entire surface of this phase image, you get something that's only a quadratic function. You get something that's pretty much constant, as long as the water volume exchange is small. This is the concept of dry mass, which is, we didn't invent that. This came about in the 50s when people started to use the phase contrast microscope. They realized there's some relation between the density of the cell and the phase contrast image. But I think we're the ones to show mathematically or physically where that is coming from. So long story short, a few years ago we were able to basically weigh cells like this just by imaging them. So this is looking at the Coli that's dividing. These are dry mass, a couple of picograms of dry mass, and then the daughter cells, they're growing. We're able to show that these are exponential growth and so on. Maybe some of you are familiar with analysis work here, where he's weighing cells with a vibrating cantilever, which is very nice, but it has the limitation that you have to keep flowing the cells through that channel, through the cantilever. In principle, it would be nice to have the ability to look at cultures of cells that are both single cells, both flowing or touching, or look at the effects of touching one another and so on. Now, what I want you to talk to you today is about something else. Now, we put a lot of work into getting very high resolution phase images, and then in order to do growth, you replace all that image of the cell with one number. So it's like kind of throwing away all that effort. But since we have all this information, so the map is essentially a density map. This is mass moving around in space and time. We're wondering whether we would be able to understand the physics of the transport just based on this data. So it's kind of an inverse problem. Can you come up with a diffusion coefficient from this data, right? And in principle, the answer should be yes. And this is just showing how much signal we have here. The slim image, we have a signal to noise of about 1,000. So it's the background is sub-nanometer, and then the highest phase we can measure is of the order of the wavelength, hundreds of nanometers. So here I'm showing that if you simply adjust the colors in this background, that looks like nothing here. Actually, there's a lot of signal, it's just the colors, the RGB cannot capture them properly. An interesting thing is that if you take the gradient of this image, you end up with something that looks like DIC, like Nomarsky microscope, and that's really confusing for the biologists when we tell them that we got the DIC image with the phase contrast optics. But basically, it's just a derivative of the phase. So once you have the phase, you can do whatever. In particular, we're able to show that, there we go, that if you take a second order derivative, if you take the Laplacian of this thing, you start to get the kind of new information. You start to see all these details that are absent in the phase. So it's a high-pass filtering, essentially, that reveals all these little details. Anyway, so there is a lot of information in there. We have this mass captured in space and time. How can we analyze to come up with some physical parameters? So back a few years ago, we came up with this idea that basically relies on calculating dispersion relation. Okay, so in the same way in optics we use it all the time. Dispersion relation in vacuum means that spatial frequency k is linear with omega. Okay, so temporal spatial frequencies are related. That's the dispersion relation. So basically we did the same thing here. We took, if you take a Fourier transform of this in space, and then you have one of those Fourier transforms at each moment in time, you can calculate the bandwidth in time and plot it at each spatial frequency qx, qy. So at each point here you have a bandwidth, okay, in radians per second. And these are the spatial frequencies qx and qy. So what we found is very interesting, that for live cells, pretty much for all the cells that we looked at, it looks always like this. This is the dispersion relation, again, delta omega, the bandwidth versus spatial frequency. It's a combination of a linear dependence with q and then a quadratic one. Now, what it means, if you look at the diffusion equation with the drift term, or the advection term, so it means that whenever you see q that is linear, that's a sign of deterministic transfer. It's a sign of a drift. It's a sign of flow, essentially. While q squared is a sign of diffusion, of a random motion. This is what you see when you do dynamic light scattering. I don't know if any of you have used dynamic light scattering. This is the term that shows up in dynamic light scattering measurement. So now, I said that qualitatively it always looks like this for all the cells. When they die, the diffusion part extends all the way down. There's no deterministic. This is what tells you that the cell is alive. This part happens when the cell is not alive as well. So what it means is that at high Q means short distances, the transport is diffusing. So it's as if the cell is very smart and is optimizing this. So at short distances, it doesn't have to consume any energy. Diffusion comes for free, Brownian motion, that gets the job done. At the moment you are looking over distances more than a micron or two, so you're looking here maybe over 5 microns. When the cell has to transport a vesicle from a nucleus far away 5 microns, diffusion is not good enough. So that's when they have to switch to this deterministic, to this directly transfer. That's when the cell consumes energy, that's when you get the ATP consuming processes in it. So this is kind of nice. Neurons, for example, they don't have much time for this at all. So the neurons are kind of extending this all the way. If you look on the dendrite, we looked at those, it's always directly transferred. So one application of this was to look at neuron differentiation. One interesting thing about label 3 imaging is that you can really monitor this so stem cell differentiation, the entire process without killing them for 2 weeks. So we did this with Steve Stice. This is a collaboration, by the way, through an SDC center that Roger Cam is the PI of, Roger Cam of MIT. So this is an SDC with MIT leading institution, Georgia Tech and us. And Steve is a collaborator from Georgia. So we wanted to use SLIM to look at these stem cells kind of differentiating. So these look nothing like neurons, right? The T equals 0. After 3 days, you start to see maybe these fine structures developing. So these future dendrites and axons starting to form, but still not really neurons-like. After about 5 days, it becomes kind of clear what's happening here, right? So these are really a lot more elongated. At the end of the 2-week process, you end up with networks. Okay, so these are neurons that are now connected with one another, and they're exchanging information. So you can monitor all these vesicles going back and forth, and so on. So basically, first of all, we do what we do all the time. We stare at these movies for hours at a time. So if you do that, you notice something very interesting. By the way, at MIT, we stared at the cell dividing for about 2 weeks. And our collaborator was like, come on, guys, this is, it's like, we saw this 200 years ago. But if you look at these clusters, what you notice is that the neurons are extending, they're growing, so these actin filaments, and then they're looking for neighbors. And at some point, they snap into place. So for example, if you look here, you will notice some of that. So they snap into place, make a connection, then they're kind of focusing their attention to the next neighbor. So this is, I think people model this with a random walk, so they're kind of randomly sampling the neighbor. And then they find a cube, extend, boom, snap in place. And we did a control experiment where we treated the cells with lithium chloride. And notice that the cluster size is much smaller. this inhibits that growth. So they cannot produce protein that allows them to shake hands. Okay, so the first thing we measured from our images was to look at the growth. So we took the total mass, total dry mass produced by that dish. And here's what we found. So there was a couple of hours here where the cells were kind of getting, I don't know, comfortable. And then there was a steep growth for a while, for about 12 hours or so. And then nothing. They flattened the growth. So this kind of matches what we witness in the movie, is that they need to grow only enough to kind of hold hands, and then they don't need to grow anymore. And this is much smaller growth for the treated cells. And then I think the most interesting part is in the dynamics. So we did that dispersion relation thing, the Q versus Q squared. And let's look at the untreated cells, so forget the treated cells for now. Untreated cells at t equals 0, so this is the continuous line. So you see, these are log-log plot, by the way. So it's almost perfectly parallel to Q squared. Nothing is perfect in biology, but pretty close, right? Now, after 24 hours, you get the dashed line. So first of all, you notice there is an X in here. So that tells you that the slope changed. So at the end of the 24 hours, you get the dashed line that's almost perfectly parallel to Q. So again, not exactly perfect, but clearly there was a slope change between diffusion to deterministic. So basically, when you put the cells in, they have nothing to do with one another. There's no correlated exchange of any kind. And after a while, they form those networks. They have to grow, then they form the networks, and then essentially they become organized. So that's what this basically we can capture. Another thing you can do is you can look at the spatial arrangement and calculate spatial correlations, which essentially has something to do with the cluster size. So basically, from 0 to 24 hours, this is actually a power spectrum. So large correlation means narrow power spectrum, right? So you notice from 0 to 24 hours, the power spectrum gets consistently down, narrower and narrower. And it's also very interesting that it's in this intermediate spatial frequency range, not at the high frequency. This is the range corresponding to the cell size. So basically, nothing happens in the cell within each cell. Everything important happens intracellular distance, so larger scales than the cell size, which makes sense. It's consistent to what we saw in the movies and so on. Okay, so what we're doing now, so again, this is unfinished business. We're trying to understand this differentiation process through growth, intracellular transport, all this dynamic stuff, but also through forces. So there's been kind of an increasing body of evidence that actually stiffness plays a role, forces, stimulation, mechanical stimulation of the cells can guide them, can cue them, and differentiate in different kind of lines. So one challenge, again, we're challenged by our collaborators. They said, we use traction force microscopy. I don't know if any of you are using that. So this idea of deformable substrates that are implanted with beads, fluorescent beads, and then you track the beads to find the displacement field and then kind of back off the force field from there. And they were complaining that it's very tedious. So you have to track the beads, you have to wash the cells sometimes to see the beads and so on. So we had this very simple idea. We said, how about instead of putting beads, we build on this idea of deformable substrates, but actually we stamp them with a grid. So you see, it's the idea of from interferometry, since we are biased for the rest of our life. So if you measure the deformation in this grid, essentially that translates into the phase. If that was an interferogram, this deformation will be the phase captured by that interferogram. You see what I mean? So basically we are applying the same phase reconstruction algorithm from DPM, from this off-axis method, except that the meaning of it now is displacement. Okay? So essentially you convert the phase, 2 pi phase corresponds to 1 wavelength of that grid. Okay, so if you have a grid of 6 microns and you measure 2 pi displacement, that means that displacement right there is 6 microns. So long story short, it's exactly, we call it, it's a mouthful, Hilbert phase. We had a microscope developed here in Spec Lab called Hilbert phase microscopy. We thought it would be nice to call it Hilbert phase dynamometry because it's basically the same idea of reconstructing the phase, except you're measuring forces and not just not doing imaging. Anyway, so from this grid, essentially do a Hilbert transform, which is a Fourier transform filtering, Fourier transform back. The phase information is essentially giving you the displacement, and this is the displacement map. So I have to say that for about a year, we worked with Alex Levine. We gave him these displacements in one direction. And he said, but now you want me to give you force fields in both directions. So this is kind of not a very well-defined problem, not very unique in terms of solutions. He tried to put all kinds of constraints, and then in the end, you know what? Why don't we do it right? So now we have two-dimensional grids. So we provide with 2D displacement, and he's able to reconstruct the forces uniquely now. So you look at very recent kind of reconstructions. This is XY displacement, and these are the forces. These lines are actually the length of the vectors, and they are pointing in various directions. And if you squint a little bit, you will see these are the slim images on top. So the beautiful thing is, the fluorescent channel, which comes in epi, in reflection, gives you the force map. And now in transmission, you do everything you were doing before. Growth, whatever. So that's what you see here on top. Probably is not the best representation, but this is just very, very recent. So basically, we would like to now quantify differences between stem cells at t equals 0, and then the various cell lines that differentiate like osteocytes, fat cells. There must be a difference, right, in their stiffness and therefore in the forces that they can apply. So finally, if I have 2 more minutes, okay, pizza's getting cold, sorry. So tomography, because we're using white light, this has a spectrum that's very, very broad, which means that the temporal correlation function is very, very narrow. And for those of you who are familiar with optical coherence tomography, developed next door right here, in Fujimoto's lab, this is a way to actually section through tissue. So it's called coherence gating. The fact that you have kind of a virtual slice through the tissue, stuff doesn't interfere unless you look at within the coherence length. So we have this effect in our slim measurement just because you're using white light, but it's also very different from OCT in other respects, in the sense that OCT can give you sectioning without high numerical aperture objectives. In our case, we work at very high numerical aperture, and that actually plays a role. The difference is basically that in OCT you have a separate reference. In our case, our reference is the light that didn't scan. So anyway, if you just scan the object through focus, and you do phase imaging using SLIM, you are able to reconstruct, this is just the raw data. So you get phases, you see stuff in focus here, top of the cell, nucleolus showing up, and then finally these things are in focus, the processes. So there's clear sectioning in there without doing anything, just taking phase imaging. On the other hand, you see that there is a lot of out of focus light that creeps in, that is kind of annoying. So essentially what it means is that we need to take this field information, this phase imaging, and do an inverse problem that will give you the structure, that will give you the scattering potential. Because so far this is light. Okay, inverse problem means solving that will give you the object. Okay, so that sounds easy enough, but it took us 2 years to actually calculate that. We didn't really know how to do it for a long time. There is a lot of scattering theories out there, particularly Wolf published this paper in the 60s, exactly based on this, how to use holographic data to get 3D reconstruction, solving inverse problems. The problem was there was a monochromatic field, and the second difference was it was a far field measurement, so it was a scattering measurement. So we had to do the calculation to generalize it for white light and bring it into the image plane. Okay, so image plane, no image plane, that's easy, but the broad spectrum part of it took us a long time. But anyway, we managed to do it. So essentially, this is the data, this is your object. Imagine this is a refracted index distribution. It's in transverse spatial coordinate, spatial frequency, and Z. So notice, the smearing in Z, the blurring, is done through this convolution operation. Okay? And basically this function, the system function, has two terms. One, due to the spectrum, so this S right here. So that tells you that the broader the spectrum, the better sectioning you have, which we know. And the second one is just numerical aperture, meaning the higher the magnification of your objective, the better you are. So that also makes sense. So notice, if you only have high numerical aperture with monochromatic light, it doesn't work. So if you use this with a laser, it doesn't work. And vice versa, if you use white light with low numerical aperture, it doesn't work. So it's a beautiful thing that they work actually together, and it's normally, if you were to make a microsensor interferometer align within the coherence length, which is 1.2 microns for white light, that would be a terrible task for grad students. Somebody elbows it, it goes away. So you have to align them within a micron, right? But in our microscope, actually, what interferes is the light that went through with the light that scattered off. So they are intrinsically in phase. And they are running out of coherence as the angles get bigger and bigger. That's kind of the idea of this formula. That the spectrum and numerical aperture are coupled. Anyway, long story short, if you start with phase images like this, the raw data, you reconstruct, you do the 3D deconvolution that I showed earlier, you start to see interesting stuff. For example, this is a Coli. You start to see this kind of helical, this is a protein distribution that some people make careers out of this thing. But I think we saw this for the first time without fluorescence. Usually, you can only see them with fluorescence tagging. In more complicated cells like this, HD29, for example, this projector doesn't do justice to our images, I don't think. Well, okay, you see some red stuff in there. So you see nucleoli, that's high refractive index. And then you see some green stuff, that's nucleus. Anyway, the whole thing started to look like a confocal image, except that this cell is not tagged, it's not fixed, it's happy, alive, and so on. So there is a lot to be improved here. We're working towards that, but I think it's very promising that we could do one day just 3D plus time imaging without killing them. So again, indefinitely. Do all that transport stuff that we're doing in full 3D. That's kind of our view. But now we got, just when we felt good about ourselves, the Nature Photonics paper, then these other collaborators said, can you do this in thick stuff, like embryos? So now, we only looked at single cells. An embryo is, imagine a layer of 10 cells. So the problem is, the optics problem is multiple scattering. So embryos or embryo bodies, these are very multiple scattering, let's say 250 microns. So we just stick that. I said, I'm an optimistic person. I said, yeah, let's just put it in there, and this is what you get. Not much. You cannot see much if you just put embryos in slim. So you see these kind of contours of the cell. They divide. Okay, so that cell, the third cell in there, but you cannot see anything else in here. So first of all, why is this important? You would like to be able to predict the viability of the embryos. Okay, these particular ones are bovine embryos. It's a big problem for cattle breeding and so on. But in vitro fertilization is actually, I think it's becoming more and more important for us humans as well. So it would be nice to, instead of implanting 8 embryos and ending up with octuplets sometimes, to be very predictive about it. So find signatures that are actually predictive of viability. Anyway, so essentially SLIM doesn't work out of the box. And the reason is that this reference field decays due to multiple scattering. Actually, this decays to 0. Your reference is very, very weak. The contrast of that interference is very, very weak. So instead what we did, we came up with this method that we call, for now, gradient interference microscopy. It's essentially what SLIM is doing for phase contrast, where you take control over that ring. GIM is doing that for DIC. So in DIC, what interferes is this image with a slightly shifted version of itself within the diffraction spot. So we control that phase difference between the two, and we end up with images like this. Okay, so now you can see these are fat droplets in each cell. This is still 250 microns, very multiple scattering, but now the two interfering beams are always equal in power. There isn't one that runs out of power, like in the SLIM. So in other words, the gradient technique is much better at kind of removing multiple scattering. So here's a version where these are hours, and now these are days. So we ran an experiment with this with 60 different embryos over a whole week, 18 terabytes. So we are sifting through all this data. So notice what happens, the sad thing here. This thing is unhappy at some point. Right about now. Boom. See? And the interesting thing to me is that when a cell breaks apart, it moves a lot, like pieces fly all over its diffusion. When this thing dies, it jellos. So it looks like the viscosity of the whole thing went up, which means that there's a lot of active processes that move these things around. I always have thought they were diffusion.
Is this a consequence of environmental changes in the medium?
To be honest, we don't know for sure. So this things happen spontaneously for some reason. It could be us, since it's our first experiment. I don't exclude that.
Is it happening to the others? Or?
No, there are some that actually go, so this is critical up to 7 days. If we can monitor them kind of predictably for 7 days,